Hello everyone, welcome to the series of videos where we are learning how to use SnapGene for cloning our own plasmid and designing our vectors for cloning experiments. So in this series, we have previously learned how we are using the restriction enzyme cloning and Gibson assembly and why we use this technique. So let's jump on PCR cloning in this video and we will learn how the PCR cloning is performed using SnapGenes and how we can efficiently use the SnapGene tool to make our own plasmid using PCR cloning and why we use the PCR cloning technique. So before going to SnapGene, I will tell you that there are two type of PCR cloning techniques as TA cloning and TOPO TA cloning. So in both of the techniques, we would need the TA overhang or we will generate the sticky end using this TA, the thymine and adenine nucleotides, and we can use this PCR cloning for making primers and generating sticky ends or adding restriction enzyme to a vector you want or to the fragment of your interest. So we can do all these things with PCR cloning. So let's get started with SnapGenes, and in this video we are going to learn how to design your vector or your plasmid from the scratch with any fragment, any gene you want and you want to add it. I'm going to teach you step by step how to do it. So let's get started with SnapGene. So that's our SnapGene homepage, and we will just click on plus for our new sequence. And I'm going to do it with new sequence. We'll retrieve it from NCBI. So just click on new sequence, the DNA file, and you will get this. This is untitled, there is no gene, but we will make a plasmid out of it. So let's just say if you have any DNA sequence already and a very specific fragment for it, you can just paste it in here. If not, what we will do, we will just go to NCBI. Okay, so let's just go to file and we would want to import a NCBI sequence. So if you have sequence already, you would put it in here. You will paste it, your sequence. If you don't have the sequence or you want a whole gene, you want to retrieve a whole gene from NCBI, you can do it by that, uh, by clicking file and importing the NCBI sequence. So we are here. You can just add your accession number here and you can click on import. So what we'll do, we will just go on our Google Chrome and we will go to NCBI website. Here we will select the database and that is the nucleotide, and you can take any example, any gene. For example, let's take human CD19. And we get it in here. Okay, so there is one more beautiful thing about SnapGene, that you can just when you are doing this, you are importing the NCBI sequence. If you have the mRNA sequence, it will convert it to double-stranded DNA. You don't need to go and check the compatibility and retrieve the other sequences. Look for the DNA for your gene. If you have it in mRNA form, that is all right. You can put it in here and then it will automatically convert it into double-stranded DNA format. So let's just again go to NCBI. Okay, right here. So let's just go to this simplest complete CDS gene of human CD19, and this is the data. I have explained everything you get in NCBI website in my other videos. You can go and understand all these things, and in this video we will just copy the accession number and the version of accession number. So we'll just copy it from here and we'll go back to our SnapGene. We'll go it like importing NCBI. We will paste the accession number and import. It will download the data with this accession number, and you can select if you want the linearized form or circular form. I will go with circular in this because it gives you the better visualization like this. This is human CD19, and you can see all the restriction sites it has. Now what we are doing is PCR cloning. Now what we want to do, we want to add a vector to this fragment, and we don't want to serve for the restriction enzyme because we are assuming that we have a different vector. So we'll just do that. This is our fragment of gene, and we will go to action here. Now what we want is PCR cloning, and we will go with TA cloning. You can go with topo cloning or TA cloning. The difference between TA cloning and topo TA cloning is just that in topo TA cloning you will also need the TA overhang, but the topoisomerase enzyme will going to be used in the topo TA cloning. So what is difference between it? In normal TA cloning, for ligation, the ligase enzyme will going to be used, and that is a common enzyme that cells use in vivo also. And topoisomerase is also a common molecular enzyme that is being used to reduce the topological uh, deformities, and in this topoisomerase will use for the ligation. It is very efficient for ligation, and when we are doing it in test tubes, when we are doing it in vitro condition, so it hardly takes 5 minutes for ligation and it's very efficient. So we rely on that enzyme a lot while cloning and other molecular experiments. So let's continue with this. We are going with TA cloning. You can also go with topo cloning. So TA cloning here. Just click on it and you will be sent to another window. Here you can see the project here. Mostly we will do it. We have the fragment, the linearized vector, and then our product. So we have selected our fragment, any gene you want, gene of your interest, you want to add it in one of the plasmid, you can do that. Or you can add one or more, you can also do that. For this example, let's go with linearized vector. So we are going to purchase it from different companies, and why and how we are going to use it in PCR cloning. So if you think about while using Taq polymerase in PCR amplification, it generally leaves a 3' end in PCR products because it frequently dissociate and then again associate, and these amplification, these replication techniques are keeps going on. So with this PCR cloning, we are going to take advantage of that extra A. We are going to get this T overhang vectors and going to pair this with this A in PCR products. So we have got our fragment. We have amplified it using PCR. Now it will be having some left A in the PCR products. So these linearized vector which has T overhang going to pair, going to bind with these fragments, and we will get the product. So to make you understand, I'm going to again show you this. This is your fragment. We are going to amplify it using PCR. Okay, now we will get the linearized vector. We are going to purchase it, and these are the shown options. You can use any of it because now we are using it just for example. If you have any specific gene and the uh, characteristics of vector you want it to be there, it will be specified for you. For example, let's say I will get this, and this is the vector, and here we are going to add our gene, the fragment we have selected. Now as you can see, it has T overhang here. But what you can't see is you don't have any sticky ends in here. So what will we do? As you can see it, you would want a 3' overhang in your gene to pair with this linearized vector. So it's very simple. Your any gene is made up of different nucleotides, and if you are talking about just one or two nucleotide, it becomes a simple task rather than finding a whole set of section with restriction enzyme and the specific compatible restriction enzyme if we use the restriction enzyme technique. So it is simpler and you can use it with any gene you want. So let's just click on add 3' overhangs to insert. This is our insert fragment. We want 3' overhang to pair with this T overhangs in the linearized vector. Just click on it, and it is ready to clone. Now we have generated the A overhang here and here. Now these are ready to pair. This will be our product. It has a linearized vector as well as our fragment of gene. Like for an example, I'm using the whole gene in here. If you want to be specific, you can choose any sequence you want in the first page I shown. So for now, let's just click on clone. And as fast as this, we have our clone DNA. It has our fragment, it has the specific characteristics of vector, and now you can save it and add another gene of your interest, another, what would you say, the specific restriction sites you want. You can add that using this technique also, and you can mix and match with different techniques if you want. You just need to save the different clones. You, if you want to add any amplification resistant sites, you can use that, and if you want to add any markers, you can add that also. So it is a simple technique you can use for 2-3 different queries, and it is simple to generate the sticky ends because you are just using few nucleotides. So I hope you understand this technique and how to use it using SnapGene. So if you like it, please like and share. You can save it from here. As you know, save as this is clone DNA, and the PCR clone product. or CD19, anything you want. You can save it in your desktop, or if you have different files, whatever, you can use it for further cloning experiments. And if you have any doubts, you can ask it in comment section. Thank you so much for watching.